Hello students, welcome to Trunkie Academy. Again today we are here for another class, okay, and today with another new topic, okay, and yesterday we studied about the sex determination in a different group of organisms, a different the way of sex determination, okay, and today we will study about the genic balance theory, okay. So yesterday we discussed that there are some mechanism in different organisms has a different name. Some are like X or XY mechanism, or Z0 or W mechanism, or ZZ or ZW mechanism. Okay, so in some cases we find that females are homogametic, like in human beings, and in some organisms females are homogametic and females are heterogametic and males are homogametic. Okay, so that those things we studied yesterday. Now, what is genic balance theory? Explains the mechanism of sex determination in Drosophila. There was a scientist who studied about the Drosophila extensively, okay, and he was also very much interested to study about the genetics and the mechanism of the transmission of characters in the offspring. Okay, so it explains the mechanism of sex determination in Drosophila by genic balance theory. It was proposed by the very famous scientist who worked. Okay, it is C.B. Bridges. Okay, his name was C.B. Bridges. Proposed this genic balance theory of determination of sex while working on Drosophila. So what is the genic balance theory? That we will go into the detail. According to this theory, sex of an individual is determined by the balance between the genes for femaleness located on the X chromosome and maleness located on the autosome. So what he concluded from his experiment is that a balance between the genes for femaleness that is located on the X chromosome, or the genes that is located on the autosome that is responsible for the maleness. Okay, so sex index is equal to number of X chromosome divided by number of sets of autosomes. So how many X chromosomes are there? Okay, if the X chromosomes are more in number, then it will be the female. Okay, then the offspring will be the female in case of it. Now that is called the X by A. So X by A, that means a number of X chromosome divided by the number of autosomes, or A. Now C.B. Bridges, he studied the offspring resulting from the non-disjunction of X chromosome during meiosis in females. Okay, non-disjunction is a kind of a phenomenon where during the meiosis the chromosomes are not separated. Okay, sometimes it happens, and this he studied about this non-disjunction of X chromosomes in some females Drosophila, and is this the failure of paired chromosomes to segregate or separate during the anaphase of first or second meiotic division, or the result is the production of aberrant gametes or with abnormal number of chromosomes. So what will happen then? So in case of female human beings also, we find sometimes that when the gametes are on the chromosomes are not able to separate, then there is abnormality. Okay, so then there will be the number of sex chromosomes will be more than the required. Okay, so this result in the abnormal number of chromosomes due to non-disjunction of the chromosomes during meiosis. Okay, not coming apart. It has to separate. Okay, or it has to segregate during the meiosis. Okay, so this will cause the non-disjunction and will be the abnormal number of chromosomes will be present. Okay, so there are various types of gametes produced. A double XX, okay, where there will be two sex chromosomes, and A0, okay, gametes which contain one extra chromosome, X chromosome, or gamete which contains one chromosome less. So there will be only autosome and there will be no X chromosome. So karyotype may be 2n plus 1, so extra. In case of this A XX, there is one extra X chromosome has come because due to non-disjunction. it has come with the gametes. So it is causing 2n plus 1 or 2n, then or 2n is the normal condition. Okay, then normal gametes aberrant. Okay, in syngamy, what will happen? Produce the fertilization. After fertilization, they go aneuploidy zygotes, aneuploid. Okay, a different, abnormal. Okay, little different. Okay, so what will happen then? Fertilization of an unusual ovum A0 of female with sperms of AX of male, and will produce male AAXX. So this will be, for example, A0 and AX sperm is the sperm AX, and then it will form AAX0, that will be the male Drosophila. It will fertilize, okay, male. So that means fertilization of unusual ovum AO of female with sperm of male, and produce AAXXY. Okay, so that means it will be the AXX, that will be the ovum, and AY sperm, that will fertilize, and then it will form male. So you can observe that when the autosomes are there, okay, and Y chromosomes are there, it will be becoming male. Okay, so it will be fertile male. Okay, and in the previous case it was the fertile female. Okay, sorry, fertile male here also, because the autosomes are more than the X chromosomes. So here also we found that the AAXX. Okay, so though this AA and XX are equal in number, but there is one Y chromosome also. So this individual will also be male, fertile male. Okay. Now, in this, it is important to note that the presence of Y chromosome in the AAXXY fly did not cause maleness, and its absence in the AX0 flies did not produce femaleness. Okay, yeah, it's a very interesting point. This is a very interesting point. AAX, it is not due to the Y, the fly was male, or it is not due to the what you call this, or absence of this did not cause femaleness. Okay, so this clicked in the mind of the C.B. Bridges scientist, and he thought, why is this so? Okay, why the presence of, it is not the Y chromosome, because even the Y is absent, then it is a male, okay, not the female. So it is then after doing research, he came to know that it is the autosomes, it is AA that is causing the maleness. Okay, so AAX0 flies, okay, and AAXXY flies. Okay, then it is the, now we will come to another point. Bridges crossed diploid females, normal diploid, okay, and he found that diploid females, okay, with three AAA and XXX. Then he found that, he obtained that normal diploid females and diploid females and intersex and super males, okay, and super females. So all these varieties he found. Okay, because due to these diploid females, there was a chance of different recombination. Okay, there was a very good chance of different recombination. So he found not only normal diploid females, but also called diploid female, intersex, super males and super females also. Okay, so and the normal males also he found. So all the different varieties of offspring he found by crossing between the diploid females and the normal diploid males. Okay, so the occurrence of diploid intersex from such a cross clearly established that autosomes also carry genes for sex. Yes, because why there are so many different varieties he got in the result? Because, and why did he got the intersex? Because it proves that the autosomes are also involved in the sex determination. So Bridges realized that the critical factor in determining the sex is the ratio of X chromosome to the number of haploid sets of autosome present. Haploid sets. If the haploid sets of autosomes are more, then it will be the male. So he concluded that Y chromosome in Drosophila lacks male determining factor TDF, like we found in the human being, or testis determining factor. okay, TDF. Okay, this is one of the important factor, a gene which causes the maleness. Okay, but this is lacking in the Y chromosome, so this is not deciding factor. So Y chromosome is not deciding factor. So it is the autosome that is also play an important role in deciding the determining the sex of the Drosophila. Is encoded by the SRY gene, sex determining region. Okay, so this means that this TDF, okay, and Y chromosome, okay, this is encoded by SRY gene, okay, gene which is responsible for TDF, testis determining factor. Okay, so that that determining factor is regulated, is expressed by the gene called SRY gene, sex determining region Y. Okay, and the Y chromosome of Drosophila is required for male fertility. Okay, so in X0 males, sperms are developed but are non-motile in condition. Okay, sperms are developed but they are not motile. Okay, so it is not normal condition. Okay. Now the Y chromosome of Drosophila is required for male fertility. I told you. Okay. Now females have a ratio equal to 1.0. That means what we call that, the number of autosomes equal to number of sex chromosome. Okay, so if the ratio is less than 1, okay, such females are called super females or meta females. Okay, super female, and they are infertile. Okay, they are super females and infertile. They will not produce the offspring. Normal males have X chromosomes to two sets of autosomes. So normal the ratio between the X chromosome to the set of autosomes, then if it is a 0.5, okay, and that means, okay, this is a little normal condition. When the ratio is more than 0.5, the males are called super males, meta males, and they are also infertile. Okay. Now conclusion by the Bridges: if the ratio is between 0.5 to 1.0, that 1 ratio, the flies are called intersex, which are larger with morphological abnormalities, rudimentary bisexual gonads. That means if they are more than 1, that is the super female. But if it is between the 0.5 to 1.0, the flies are intersex, which are larger with morphological abnormalities, and the rudimentary gonads, which are non-functional, and the offspring produced by these individual will be not fertile. They will have the very rudimentary gonads, which is not functional. Okay. Female determines located on the X chromosome, and the male determined on the autosomes. So the C.V. Bridges, he came to an idea that the autosomes are also involved in the determining the sex of the Drosophila. So he concluded that it's not only the Y chromosome which is responsible for determining the sex in the Drosophila, but it is the autosome also, because the ratio of these sex chromosome and the autosomes are together. Then it decided whether the individual will be the male or normal male or super males or super females, depending on the ratio of the sex chromosome and the autosomes. Okay, so this is the genetic balance theory actually. So he formulated one summary chart showing different ratio of X chromosomes and a set of autosomes controlling the sex in Drosophila. There's X by A ratio. Okay, so if it is less than 1.0 and 1, then it will be super female, and if it is 1.0 equal to 1, then it will be female, and if it is 1 to half or 0.5, then it will be intersex, and if it is 0.5, then it will be male, and 0.5 less than more than 0.5, then it will be a super male. And these super females or super males will be infertile. They will not produce fertile. They're not going to produce the offspring. Okay, viable offspring. Now, Bridges' genetic balance theory, example Drosophila. So the super female, then they have got the 3 X chromosomes, and set of chromosomes autosomes are to AA, and the sex index ratio will be 1.5, like it's more than 1, so it is super female. Female, the tetraploid, where we will find the 4 X chromosomes and 4 set of here it will be is equal to 1. So here it will be the this will be the female tetraploid, and in case of female triploid, triploid, there will be 3 3. So in both the cases, it is ratio is 1, so it is the female. Okay. Now in case of the female diploid also, it is, since the ratio is 1 in case of X chromosome and 2 sets of autosomes, so here it will be the ratio will be again 1, and this will be the female. Okay. And intersex it will be then it will have the 2 X chromosome and 3 sets of autosomes. Then it will be little more than the half, or 0.50. So it is intersex. And in case of the triploid, 3 sets of 3 X chromosome and 4 sets of autosome, then again it will, the ratio is going to be 0.67, which is also the intersex. Okay. Then in number 4th case, it will be normal male, where there will be one X chromosome and 2 sets of autosomes. So that will be equivalent to 0.50, and then this will be a normal male. Then super male will be X, and then there will be 3 sets of autosome. Then it will be less than 0.50, less than half. Then it becomes the super male. Okay. Now next is the female XX, and then you see this is the chromosome of the Drosophila. Okay. A Y chromosome little bent in that region, in one terminal region, you can see. So that is representing the Y chromosome. Okay. And here fertile male XY Drosophila chromosome, and here is the, now this, okay, sterile male will have that means XY. Okay. Sorry, the Y will be the missing. Okay. So there has to be XY in normal condition. Okay. Now we'll come to the MCQ. Bridges concluded that Y chromosome Drosophila lacks genetic information essential to male fertility, or alleles specifying traits, or male determining factor, or all of these is male determining factor. Because in case of the human beings, SRY gene, a gene which is present on the Y chromosome, that is responsible for producing a factor called TDF, testis determining factor, which is lacking in case of the Drosophila. Then he came to conclusion that, so it's not the Y chromosome, it's not involved in the deciding the maleness of the offspring that is produced. So it may be some other reasons. So after doing lots of research and experiments on the Drosophila, then he came to a conclusion that this is not the Y chromosome, because the SRY gene, which is responsible for TDF, is not present. The gene is present, or may be present, but the TDF is not there. Testing determining factor is not present, so it's not the Y chromosome. It is the ratio between the autosome and the sex chromosome. In Drosophila, normal females have a ratio equal to 1.0, 0.5, 1.5, or 0.6. It is 1.0. Yes, for the female normal, it is, we saw that whether it is a 4 sets. Again, if the ratio is equal to 1, and we came to we saw that it will be what we call this normal female. Okay. And if it is more than 1, then it will be a super female. Genic balance theory, ratio of sex determination is applicable to all XXXY methods of sex in animals, or Drosophila, Femia, or birds. No, it's not all. It will be applicable to Drosophila. This has been found in Drosophila, because now this is important for NEET examination. Please keep in mind that this theory, this genic balance theory, after doing so many experimental researches, he found this. Okay, there is the involvement of autosomes also in determining the maleness of the offspring, but this is not the case in case of the human beings. Why? Because there is a clear-cut, a factor called TDF is present, which is present on the which is encoded by the SRY gene. Okay, that is present in the Y chromosome. So this SRY gene is responsible for encoding the TDF factor. So this TDF is there. So there is no question of genic balance in case of all XX or XY methods of sex determination in animals. Okay. So this is applicable to Drosophila, not for all XX or XY methods of sex determination. Okay. In Drosophila, AA or X flies, these are the fertile males, or sterile males, or sterile males, or intersex. So this will be sterile males. Okay, males, it will be male, but they will be sterile. They will not going to produce the okay. Now in case of the bar, now this is one very interesting thing. Okay, so later when the so many research, even now also so many scientific and biological and some medical researches are going on every always it's an ongoing process. And when as the technology is advanced nowadays, okay, and we have come to know that so many things, so many new things are discovered about the different phenomena and different processes taking place, whether it is about the transmission of characters of from parents to the offspring, or it is the different types of physiological process taking place in our body. So Barr body is another important structure that we'll study, which was found after some research by the scientists, and this is little surprising and little unique. Okay, who discovered first? It was the Barr and Bertram observed the chromatin bodies. So it is a chromatin body. Chromatin is the chromatin fiber that is present in the chromosome. Okay, when it condensed, it formed accumulated like a structure. Then it is called a Barr. Okay, and the name of the scientists of the Barr and the Bertram, they both of them they observed these chromatin bodies. Where they observed, we'll come to another. Murray L. Barr. His name was the Murray L. Barr, and present in the interface nerve cells of female cat. They found this Murray L. Barr. Murray Barr, he found this in the interface. Interface is the one of the stage of the life. What we call the cell cycle. Okay, so in the cell cycle of one of the stages of the cell cycle, that is the interface of the nerve cell, they found these Barr bodies. This was by the Murray L. Barr. The name of the scientist is Murray Barr. Okay, and these bodies appear as a darkly stained body attached to the nuclear membrane. So they were surprised. What is it darkly attached, highly stained, darkly stained, little easily distinguishable and distinct, prominent structure that is attached to the nuclear membrane. So then after doing research, what are these bodies? They started thinking, and after research they came to know heterochromatinized X chromosome, heterochromatinized. So they came to in humans, this chromatin body in the female cells are derived from the buccal mucosa, or in fibroblast. Buccal mucosa means the tissue that is present in our mouth or buccal cavity. Okay, the layer. Now the undifferentiated connective tissues. Okay, fibroblast is the undifferentiated connective tissue cells. Okay, then the cheek cells. And these the buccal mucosa is the cheek cells, or fibroblast. Okay. Now, a Barr body appear on one of the lobes of some neutrophils in females. Neutrophils is a kind of white blood cells. Okay, there are eosinophil, neutrophils. Okay, and white blood cells, different types of white blood cells are there in our body. And in neutrophil, in females, this is appears as a Barr body, and which shape? Okay, a small drumstick like projection, drumstick. Okay, so they are drumstick, two drumstick. So they small in one terminal region, it will be accumulated like ball like, and there will be some elongated structure. Drumstick like projections are found. And this, what is the importance of this technique? The sex of the human embryos can be distinguished at early stage of development. So by knowing this, okay, by taking the neutrophil, okay, and we can come to know the sex of the embryo that is developing. Okay, in which organism these cellular characteristics are generally seen. So this is seen in all mammals. Okay, so Barr body are present in all. Now coming to the leonization. Inactivation of one, now this leonization and Barr bodies are interrelated. Now after studying this leonization only, you will be able to understand, completely able to understand what is Barr body. So what is this Barr bodies actually? The leonization, when nearest means inactivation of one of the two copies of X chromosome that is present in the female mammals. Okay, there are two X chromosomes. Okay, during the formation of gamete, when the meiosis takes place. Okay, the ovum cell or egg cell will contain only one set, haploid number of chromosomes, n number of chromosomes, and along with the one X chromosomes. During the fertilization, after the formation of zygote, when there will be the two sets, the diploid number of chromosomes, then there will be two X chromosomes. When the zygote will be developed, there will be rapid cell division, embryo will develop, there will be implantation, the fetus will be fully developed. Okay, then the delivery of the child will take place, and then this will be, then there will be inactivation. Okay, this when absorbed in these cells, okay, it was found that one of the two X chromosome was inactive. Okay, inactivation of one X chromosome present in the cells of the female mammals, that is called leonization. So that looked like a bar. So that's called a bar body. Makes the inactive X chromosome transcriptionally inactive. So for a chromosome to be active, to remain active, there should be transcription and then translation. So this process should continue because for that particular chromosomes and the cells to remain alive, okay, and to be effective and functional. But here, since the leonization is taking place, this leonization means inactive of one of the X chromosomes, that is causing the that is caused due to inactivity of the transcription process. So transcription is inactive and silenced, so there is no this chromosome is no longer functional. Okay. Now this structure is called heterochromatic body. So dosage compensation. Female mammals have two X chromosomes in each cell. X inactivation in their cell causes them to not to have twice as many products of the X chromosomal genes. So that means the genes that is present on the X chromosomes, that will be 2 in number. There will be 2 copies, because since the genes present on both the X chromosomes are same. So if the both are active, then the number of genes present on both the X chromosome, all the genes will be 2 in number, 2 copies. So make sure that there will be only 1 copy or only single copies of each of the chromosomes that is present in the X chromosome. One is becoming inactive, and this is called leonization. So as those of the male possess only 1 X chromosome in each cell, so that is going to have only 1 copy of the genes that is present on the X chromosome. Because if there are 2 X chromosomes, then there will be 2 sets of genes that is present on the X chromosomes, which is not required in the human body or all other mammals. Okay. Now, regardless of how many X chromosomes a somatic cell possesses, all except one of them appears to be inactivated and can be seen as bar bodies. Now you are getting a clear idea and clear picture about what is bar body. Now this leonization and this inactivation of 1 X chromosomes, because only 1 copies of genes are required from the X chromosomes, this is going to change into an inactive form, and that appears as a bar body, and is called bar body, named in the name of the scientist called Murray Bar. Okay, so no bar body is seen in the somatic cells of Turner's females, because since they do not have 2X, because this is the abnormality. The Turner syndromes and Klinefelter syndrome is a kind of abnormality during the due to non-disjunction, during when the chromosomes are unable to separate or segregate during the process of meiosis. So this kind of abnormality is caused, and we find that the individual body, that the somatic cells will contain 1 chromosome less than the normal number of the chromosome. That is, this will have the 45 number of chromosomes. Though there are normal number of chromosomes is the 46. Since there is only there is 45 chromosomes, that is 1 chromosome is missing. That is only 1 X chromosome is there. So we do not find bar bodies because only 1 X chromosome is there. 1 bar body is seen in Klinefelters. Okay, because there is 2 X chromosome and Y chromosomes, because it's only 2 X chromosomes. If there are 3 X chromosomes, then there may be 2 bar bodies. But here we find only 1 bar bodies in the Klinefelter syndrome, because there is 2 X. Because here this is also an abnormality. We find the 1 chromosome is extra. It is more than the 46. Here the 47 chromosomes are there. So 2 bar bodies are seen in case of the females with 3 X chromosomes. Okay, because only 1 X chromosomes is sufficient to encode for the genes, okay, that is there in the X chromosomes. Okay, so no duplicate copy is required. That is the reason leonization or inactivation takes place. Because now these 3 bar bodies are found in case of the individuals having 48 chromosomes, 2 extra chromosomes. That will, with the 2 extra X chromosomes, then there will be 3 bar bodies, and so on. Okay, so dosage compensation. Therefore, the number of bar bodies follow an N1 rule. So N minus 1 rule. So only 1 is required, and rest other will form the bar bodies, and that due to the leonization. Okay, so N is the total number of X chromosome, and N minus 1. So number of bar bodies is equal to the total number of X chromosome minus 1. So this is very simple. Now you have understood. Now bar body in a neutrophil, this was observed during the research and the different experiments carried out with the cells of the neutrophils. It was observed on the microscope that this structure appeared near the nuclear membrane, and when studied in detail, it was found that this is the bar body, the inactivated X chromosome, which has changed as the bar body, and which is, because it is not required, so as to ensure that there should be no extra copy of the X genes that is already present in another X chromosome. So this leonization or inactivation takes place. So bar bodies means they now, so zygote will form, random inactivation will take place. One of the two will take place, whether it is the any one of the two will take place, and then there will be the bar body formation. Okay, so this goes to the nuclear membrane attached there and looks like a drumstick. Okay, and this is called bar bodies. So this was observed during the experiment and the research while they studied about this. Okay, so this is called the bar body formation or leonization. Okay, now nucleus, cytoplasm. Okay, 46 chromosome means we have got 23 pairs. Okay, so bar body is formed and near the nuclear membrane. So this you can see here 46. So there is no bar body. Okay, and in case of the 47, there are 48. Okay, so there will be the two bar bodies, and here in case of this n-1, so this is a 3 in case of 48, and 49 you can see there are 3 bar bodies, because 3 are inactivated. This will form there are 3 extra X chromosome, so there has to be 3 bar bodies due to inactivation. Okay, so buccal epithelium means the epithelium that is present on our buccal cavity, mouth, the cheek cells that is present in the inside of our buccal cavity. So that can be easily taken out and can be observed under the microscope after staining with some special dye, and we see that the polymorphonuclear leukocytes that is also observed in the polymorphonucleocytes, leukocyte, that is a kind of white blood cells, leukocytes, where we find this kind of structure very clearly. Okay, and now we will go through some questions. Okay, now non-disjunction is the failure of separation of linked genes, or it is the failure of independent assortment of non-allelic genes, or the failure of the paired chromosomes to separate in meiosis, or it is the failure of recombination of non-linked genes. Okay, it is the failure of the separation of the paired chromosomes to separate in meiosis. Normally, in the normal process, the chromosomes should separate. They should segregate in the process of meiosis, so that n number of haploid number of chromosomes are ensured in the gametes. If this is not happening, if this non-disjunction occurs and there is no separation, then there will be extra number of chromosomes in the gamete, and then that would lead to abnormality in the zygote, and the individual will form abnormal characteristic features in the body. So this is the failure of paired chromosomes to separate in meiosis. Now abnormal gametes are produced due to the non-disjunction of paired chromosomes in mitosis, or non-disjunction of paired chromosomes in meiosis, or disjunction of paired chromosomes in meiosis, or disjunction of paired chromosomes in mitosis. This is the non-disjunction of paired chromosomes in meiosis. Okay, the meiosis is the process that takes place in the gonads. Okay, all other body parts are called the cells that is present in the body parts, muscles, skin, glands, alimentary canal, gut, or liver, gland, all organ. It is the somatic cells where the number of chromosomes are in pairs, that is the 46 number of chromosomes are there. But in the gonads, okay, in spermatozoa and egg cell, the number of chromosomes after the formation of the gametes, these are 23 in numbers. Okay, a single pair. Okay, but these single pairs are formed due to the process of meiosis, the meiotic cell division. So this is the non-disjunction paired chromosomes in meiosis. Okay, so this will correct. Now Keith Moore and the Barr in human beings studied the heterochromatization of Y chromosomes, or the heterochromatization of X chromosome, or heterochromatization of autosomes, or heterochromatization of X and Y chromosomes. So they studied all heterochromatization of X chromosomes, not X and Y, or not of autosomes, but heterochromatization of X chromosome only. So they were the scientists who first studied about the heterochromatization of the X chromosome. A darkly stained body in the interphase. So for the first time, it was observed in the nerve cell when they were observing, and they were doing some experiment with the nerve cells. Okay, and they realized that some kind of darkly stained structure is there near to the nuclear membrane, and then they come to know that this is the Barr body. So they who observed that is a Barr and Bertram or Keith Moore or Barr or Barnatine, okay, or Barr Levin. This is the Barr and Murray Barr and Bertram. Okay, they were the scientists who for the first time observed, and they started researching, and they came to know this, and this was named as Barr body. So Barr body is heterochromatized Y chromosome or heterochromatized autosome or heterochromatized X chromosome or X and Y. It is simply number C, heterochromatized X chromosome. Okay, so now in the next class we will study about the haplodiploidy. Okay, haploploidy. Now what did we study today is that first we started with the genic balance theory, and genic balance theory was given by the C.B. Bridges, scientist called C.B. Bridges. He first started with the experiment with Drosophila, and he did some extensive research and experiments with the Drosophila flies, and he came to know that it's not only the Y chromosome, it's not the Y chromosome that is deciding factor for the maleness in the Drosophila. And after doing several experiments, he realized and come to a conclusion that it is the autosomes that is involved in the maleness of the Drosophila. And after that, we studied about the Barr bodies. A Barr body is a kind of heterochromatization, inactivation of one extra chromosome, X chromosome, which is there in case of the female body. Because in the females, there are two X chromosomes, which is actually not required. Although it is required for the determination of the sex of the child, but it's not required in an individual body. The X chromosome, the genes that is present will be double in copy, and that is not required by the individual body. So that becomes inactive. So that forms a Barr body, and this was carried by the scientist Murray Barr and Bertram. Okay, and after that we studied about the Leonization, and Leonization is the process which causes the inactivation of one of the two copies of the X chromosome that is present in the female mammals. It takes place in all the mammals, not only in human beings. So this is a kind of unique features. Okay, unlike Drosophila, in case of human beings, the Y chromosome is completely involved in the determination of sex of the child, because the SRY gene on the Y chromosome is responsible for TDF, testis determining factor, and which is ultimately determines the sex of the child during the development of the embryo. So that means in case of the determination of sex in the Drosophila and in mammals is completely different. Okay, so the mode of the determination of sex that was in the Drosophila was given by the genic balance theory, which is studied by the scientist called C.B. Bridges. Okay, so after that we studied about the dosage compensation, and we came to know that if there are three leaving one X chromosome, all other X chromosome will be inactive, and like it has been observed in case of the abnormal individuals like Turner syndromes or Klinefelter syndromes, and even individual with more than 3 X chromosomes, it was found that except one X chromosome, all other X chromosomes were inactivated. Okay, so that is called dosage compensation. So all these things we studied about the dosage compensation, Leonization, Barr bodies, and genic balance theory. So tomorrow again we will meet at the same time. Okay, and right after this class, as we are sending every day DPP or daily practice paper, what we have to do is you have to go to your academic booster. Okay, and you have to log into your academic booster and you click on the resources. You'll find that there is a link called writing material. Okay, and clicking on the writing material, you'll get the DPP. So download that DPP, daily practice paper, and you solve the questions that is there in the DPP and send back to us for evaluation. We'll check and then we'll send you the remarks. Okay, so thank you for joining and listening today. Tomorrow again we'll meet at the same time. Till then, goodbye.